Thank you. Thank you so much for the opportunity to speak here. And I'll be telling you about 3D spatial navigation in Egyptian fruit bats. But I first want to explain that this is part of a larger project. Here's my research team working on this, and the postdoc, Nikita, it's not, oh, yeah. Here has really been involved very much in all aspects of the project. Keegan Eveland, Xiaoyan Yin, Shivam Chitnas, Grace Capshaw, Alara, afraid I can't pronounce her last name very well, and Magdiel. They've all contributed in other ways to this project, but it is part of a larger HFSP research project, which is led by Magnus Wahlberg in Denmark. Another PI is Herbert Hermans in Belgium, and August von Bayern in Germany. And the title of our project is Mental 3D Space-Time Travel in Fission-Fusion Animal Societies. I thought maybe I should just try to unpack that a little bit. So the animals that we're studying in this project are marine mammals, birds, and bats. So these are all animals that navigate 3D space, either in air or underwater. Fission-fusion animal societies refer to animal societies that sometimes come together in large groups and work in collectives and sometimes move apart. And the mental time travel really refers to episodic memory. And it's been assumed that episodic memory abilities separate humans from other animals. And our project aims to deepen our understanding of mental time travel, perhaps reveal that some animal species are capable of episodic memory-like behaviors. And so we'll be focusing on bats, dolphins, and parrots, combined with tests of emerging principles in robotic platforms. So let's just talk a little bit more about episodic memory. Let me give you an example. So I'll start by asking you to imagine that you're at the steps of the Peabody Library. This is a beautiful library at Johns Hopkins University. And you've been to this library the week before to look for a book. In the interior of the library, it is quite extraordinary, and you see all these stacks here. Could be quite difficult to find a book. But you seek the assistance of a librarian who helps you find the location of the book. And you return a week later, and you know exactly where to find that book because you can remember where you were when you heard about how to find the book, and you can find your way to the stack. And so the memory from your prior experience interacting with the librarian allows you to navigate to the location of the book much more easily than if you had to search through all the call numbers of the books and all the stacks. Okay, so now, do animals rely on episodic memory to carry out natural behaviors? And here I'll be talking about echolocating bats, and I'd just like to say a little bit about why echolocating bats may be useful models for studying episodic memory. First of all, bats are mammals, and they show many survival behaviors that are exhibited by other mammalian species, including humans. And I want to also point out the importance, the value of comparative studies, because they help us separate specializations from general principles. If you identify a result from a study in one species, you can only know if it's specific to that species or general if you compare the findings with other organisms. And finally, the bat's active sensing system, and that is echolocation, which I'll get into in a bit more detail, provides us with a direct window to the information the bat uses to guide its behavior. So now, let's say a little bit about echolocation. Bats use very high-frequency sounds to navigate the environment. And here is a quick video of a bat going after an insect in complete darkness. Videos together by my former postdoc, Aaron Corcoran. Now, the sounds that are produced by the bat are all above our range of hearing. It's typically referred to as ultrasonic signals. but it's been slowed down so that you can actually hear them. And so these sounds are very well suited for localizing very small objects. So a bat will produce very high frequency sounds that are well suited to reflect off of objects in the path of the sound beam. Echoes return to the bat's ears. The bat can process information in the echoes to localize the object in 3 dimensions, the horizontal plane, the vertical plane, and the range axis. And the bat may also be able to use the echo information to glean some features of the object. So this active sensing is important in the lives of bats, and we can use the echolocation behavior to understand what information bats have used to extract signals from the environment. So now, in the talk today, I'll be focusing on one species of bat, the Egyptian fruit bat. And I should add that there are actually roughly 1,400 species of bats, representing more than 20% of all mammalian species. And about 200 of these 1,400 bat species don't even use echolocation. They rely on vision. The Egyptian fruit bat does have well-developed vision. You see its nice large eyes. And it looks like he's smiling there. But in fact, these bats sort of clench their teeth and use their tongue to produce echolocation sounds. And so that the sound is transmitted through the teeth. And there are some interesting directional characteristics of these signals that I'll talk about briefly. And the Egyptian fruit bat is well known to navigate over very large ranges. So here you see a map of ranges traveled by Egyptian fruit bats in Israel. This comes from one of my HFSP colleagues, Yossi Yovel. You see the scale here is 10 kilometers. So these animals often fly many kilometers each night to find their fruit. So, as I mentioned, Egyptian fruit bats produce tongue clicks, and here is a video clip of a bat, an Egyptian fruit bat, producing these tongue clicks. These are also ultrasonic. So the sounds produced by the first bat I showed were produced by the vocal cords, but these ultrasonic signals are produced by the bat's tongue. And so by slowing everything down, we can hear the sounds. And these sounds have interesting directional characteristics, which I'll show you now here in the next slide. So what you'll see is a raw video clip of a bat flying to a perch and an animation of the same trial where we take the recordings from a microphone array. Each of these circles around the perimeter represent microphones that are used to capture the bat's signals. And with the distribution of sound energy around the room, we can reconstruct the sound beam pattern as the bat flies. So you'll see an animation of the bat flying to the perch and the echolocation signals it's producing along the way. You see. So this bat produces sounds in pairs, and each sound in the pair is directed off to one side. So right, left, left, right, right, left, and so on. Okay. Again, these are not sounds that you could hear with your ears, but with recording equipment, we can slow things down and you can hear them. Okay, so now to the specific questions of our research, and this is work that was done by Nikita, Xiaoyan, and Keegan. And the first question is, to what extent do Egyptian fruit bats rely on memory to navigate? And second, does spatial memory sometimes overshadow sensory information? So in this experiment, we had the bat perform a very simple task, and that was simply to fly to a perch. These bats like to rest, so that's their big reward. Sometimes we give them a little treat when they land on the perch, but really just the resting is all they need. And here are microphones on the walls and high-speed cameras to track the bat's flight trajectory. And we ran experiments with several trials, 10 to 12 trials in a row with the perch in one fixed position. And then we moved it around to the side, up, down, off to the side by 15 centimeters, and then 30 centimeters. Okay, so what did they do? Well, we looked at the bats' landing performance in the light and the dark. And what we found is that when the bats flew to the landing perch in the light, they performed very well. So this baseline refers to the first 10 to 12 trials we ran. And if we moved the perch 15 centimeters in the light, the bat's performance dropped a little bit. But when we moved it 30 centimeters, and it's not that far, the bat's landing performance dropped dramatically. Now, in the light, the bats can use vision and echolocation. They can see where the perch has moved to, but for some reason, they don't. In the dark, their performance is lower compared to the light. Here, they're using just echolocation. And when the perch is moved 15 centimeters in the dark, performance drops further. So let's look at the echolocation behavior of bats as they perform this task. So first is a baseline trial, where each of the clicks in the pair is represented with a stick here, and this is the location of the perch. And then when we move to the trial in which the perch has been moved 30 centimeters, this is now the perch location. This is where the perch was previously. You can see that the bat directs the sound at the location where the perch was previously. We'll do another loop around. See when it comes back. Does the same thing. It looks at where the perch was previously. So this is in the light. It can see. It can use echolocation. It doesn't find the perch. This was a big surprise to us. So this is just to show you, still frame what I just showed you when the bat was released and flew to a platform at a location that was trained on for 10 to 12 trials. It just goes straight there. But then when the perch is moved, the bat doesn't find it. So this, as I said, was quite a surprise to us. And it shows that in some cases, spatial memory can actually eclipse the use of sensory information in a navigation task. So this is funded by HFSP, and HFSP encourages following up on interesting results, even if they're maybe a little bit off topic. We decided to probe deeper into the bat's use of sensory information to guide its navigation. So just to remind you, it has excellent night vision, it has tongue-click echolocation, and we did some experiments where we asked, where do bats fly when visual and echolocation information conflicts? And this is a project carried out by Nikita Finger and Shivam Chitnas, a graduate student. And I'll tell you what I mean by conflicting information in just a minute. And if visual images are displaced, we asked, do bats rely on echolocation? Do they shift to use echolocation to guide navigation? So this is how we introduce sensory conflict. We fit our Egyptian fruit bats with lovely goggles and place different kinds of lenses in the goggles. This shows you the effect of a prism where the image is displaced. Okay. So we ask, where do Egyptian fruit bats fly when echo and visual information conflict? So, as I mentioned, we used different lenses in the goggles that the bats wore. We used clear lenses so the bats could just see through and operate as normal. We also placed dark lenses so the bats couldn't see, but they could still echolocate. We used right displacing prisms, displaced the images 23 degrees or 11 degrees, and left displacing prisms. So same, very simple task. Bat, perch, but now the bat is wearing goggles. And we just record the bat's echolocation behavior and flight trajectory as it performs in this task. So the prisms introduce conflict between the visual and echolocation information. So let's look at a trial. Here's a bat flying to the perch with the clear glass in the goggles. No problem. Left displacing prism. Here the bat has a little trouble, but manages to land. Okay. Right displacing prism. Missed it that time. Okay. And then finally, this shows the dark glasses. Here, so the bat can use echolocation, but not vision. It flies much more slowly, but it lands accurately on the perch. Okay. So here are some data from one bat that show its flight trajectories across trials when it's fitted with goggles carrying the right shifted prisms 23 degrees and 11 degrees. 0 marks the location of the perch, and here we have successive trial numbers. So initially, these gray bars represent the flight behavior of the bat in baseline trials. And then when fitted with the prisms, the bats are flying off to the side, which is what you saw in the raw video. And then over repeated trials, the bat begins to land more successfully on the perch in spite of wearing the prism. And here you see when the prism shifts the image 11 degrees, the magnitude of the bat's flight offset is reduced, and then it catches up, it figures out how to fly. Okay. So we can ask, do the bats overcome visual image displacement using echolocation? So if you or I were to wear prisms like this and were to reach for a coffee cup, initially we would miss. And then over repeated reaches, we would make corrections and we would find our way to the coffee cup. So it's sort of a sensory motor adjustment. In the case of the bat, it has a secondary sensory system it can rely on that provides the ground truth of where the perch is. So the question is, does the bat use echolocation to find the perch when it discovers that vision is not reliable? So how do we do this? Alara, who's an undergraduate in the lab, had the idea that we could fit ear molds into the bat, so the bat wouldn't hear the echoes that return to its ears. So we ran some experiments with ear molds alone, and then we combined ear molds with prisms. Now we needed to make sure first that the ear molds actually blocked echo information. So Grace Capshaw, a postdoc in the lab, did auditory brainstem response measurements to sound with and without the ear molds. And auditory brainstem responses are non-invasive measures of hearing. And so one can place electrodes under the skin and measure activity in the auditory nerve, the lower brainstem, and up through the midbrain in response to sound. So what you see here are auditory brainstem responses to sound at different levels. So 90 decibels, very loud, very big responses. The details aren't really so important here. 80 decibels, 70 decibels, 60 decibels, you see the response begins to get smaller as the sound level decreases, And right around here, we call it the threshold. Sound levels below 30 decibels aren't really eliciting any kind of measurable auditory brainstem response. When the ears are plugged, we see a different pattern where the very low levels, even up through 60 decibels, there's really not much of a response. And so the threshold is determined to be somewhere just below 70 decibels. So we see that the earplugs do attenuate the sound roughly by 40 decibels. So we know that they work, and they'll influence how well the bat can hear echoes from its calls. So we ask, where do bats fly when visual images are shifted and echolocation is disabled? So here we have the clear glass with the ear mold. So the bat can use vision, but not echolocation. No problem. Here again is the bat wearing the prisms, shifting the image to the right, but no ear molds, manages to land just fine. And now here, right shifted and ear molds. So prisms and ear molds, and this is not slowed down, because we wanted to give you the feel for how much the bat just flew around and around and around. It just couldn't land. Okay. Which suggests that in these trials where the bat did adapt to the prisms, it was shifting its reliance to echolocation. Okay, so here's what I just showed you with echo blocking only, that is ear molds alone. The bat can fly straight to the perch, but when the bat wears prisms and has ear molds blocking echoes, the flight paths are disorganized and it has a lot of trouble landing. So what are the conclusions of this work? First of all, we've learned that Egyptian fruit bats rely very heavily on spatial memory to navigate, even when visual and echo information are available. So when the landing perch is moved, the bats inspect the location where the perch was previously and often fail to land. That was really quite a surprise to us. When vision and echolocation conflict, Egyptian fruit bats are initially guided by vision. But over repeated trials, they learn to fly more directly to the landing perch, and we believe they do this by shifting to the reliance on echolocation. And more broadly, we feel through this work, we can learn general principles of spatial navigation and memory, which lay the foundation for understanding how animals plan their routes in natural environments. And we feel that these findings do have broader implications for establishing understanding of the mechanisms that support episodic memory in animals. And with that, I'd like to, again, thank HFSP for this support and my colleagues who participate in the larger project, and I'd be happy to answer any questions.
Thank you, Cynthia. We have time for questions. Oh, I see a lot of hands raising. Let's start on the right-hand side first, and then we move left. Just behind you.
Hi. Thanks for the talk. So I was wondering about whether these fruit bats live in dense social colonies, and if they do, does that influence the trade-off between echolocation, which obviously might be there might be crosstalk between different individuals versus sight and spatial memory.
So I'm sorry, you were first asking about the social colony?
So if they live in dense colonies, because I know a lot of bats do live in very dense populations, right?
Yes. So Egyptian fruit bats do live in large groups where they roost, and they may rely very much on spatial memory to find their way out of the roost. And then when they're navigating, I think they rely heavily on vision. Vision is a sense that provides information over longer distances. So even if they're exiting at dusk, there's still light for them to find their way. They can still use echolocation as supplemental information, but as I said, by and large, they're relying, we think, on vision to navigate.
Moving to the right now, there.
Okay, thank you. Super interesting talk and super interesting project. I was wondering, what would you expect if you would do the same experiments with echolocating bats, so microbats, not fruit bats?
Yeah, so no bats are blind, and so some echolocating bats rely much less on vision, but they do use vision to some extent when it's available. And so I think if we put prisms on the echolocating bats that they normally would forage for insects that rely more heavily on their echolocation, it probably would have a less pronounced effect.
May I ask another question? Did you think about playback experiments like phantom echo experiments to make a phantom target?
So have we done that, or I'm sorry?
Did you think about it?
Yeah. Yes, I've thought about it a lot. I've done many experiments with playbacks where bats are trained to rest on a perch. Their calls are picked up by microphones, digitized, delayed, and played back through loudspeakers to simulate echoes from objects. And I, many years ago, started to do an experiment where I was trying to create more of a natural, dynamic, acoustic scene using a playback system. And I came to realize that this was comparable to a human wearing sort of virtual reality goggles with a very narrow little window, and the rest of the world is behind them. So basically the salience of that virtual reality is diminished, and it just doesn't really work. It's very hard to create a virtual sonar environment because of the directionality of the sounds, the equipment that's needed. Unless you fit the bats with earplugs, then there are going to be echoes from other objects that will interfere with the playback echoes.
Next one here.
Thank you for the fascinating talk. When you showed this prism glass, I had to think of these goggles that you can get that flip your vision, and after some time you would adapt and like your brain would re-flip it back. Do you think that could also happen with the bats with the shifted vision, that at some point when you keep them on for, I don't know, a day, they will like readjust their perception?
Quite possibly. So these experiments ran for about 30 minutes for each bat. We did not have them wearing the prisms for any extended period of time. So when the prisms were removed, we didn't see any evidence of an after effect, which you would see like in a human who had adjusted to long-term wearing of images that are displaced or flipped. So we don't know.
Final question.
I was wondering if you looked into what elements of the room the bat is remembering when you have the moved perch experiment. So if you like move other pieces of the room too, will it remember better?
Yeah, so we're actually working on that. So we did an experiment where we rotated the location of the perch and the release point, and depending on the magnitude of the sort of movement of the two, we found a lot of times the bats were really confused. And we came to see that there were different elements in the room that were perhaps providing references for the bats to use for guiding their navigation. So now we've set up a simpler arena, which is curtain of felt, so with a uniform room, and then we have the landing perch and a landmark that's adjacent to it, and we're moving them together and we're moving them apart to try to get at that very question.
Okay, great. Let's thank Cynthia once more again, please. Thank you.